Welcome to this podcast on drug solubility as part of the medicinal chemistry course, the North Carolina School of Science and Math online program. Okay, what we want to talk about in this podcast is a little bit more about drug structure with a specific focus on drug solubility. If you look at this slide here, which represents a cartoon of good medicines, there are 4 or 5 very important considerations that need to be made in designing a drug. Certainly at the very top, activity, how well the drug candidate binds to its target and generates the desired biological response. In this case, curing whatever disease we're trying to cure. Metabolic profile and toxicity, this really talks to side effects. Half-life, how long the drug will stay active in its active form in the body. Oral bioavailability, how much the drug candidate reaches the appropriate tissue in the active form, specifically when given orally. And the topic of this discussion, solubility, is a discussion of how well the drug candidate can be absorbed by the body if taken orally. So this is where we're going. We're really going to look at solubility in this particular podcast. Okay, drug solubility. As you are beginning to see, and we'll certainly see a great deal more of, the guiding principle of chemistry is what's known as ADMET or ADMET. Okay, A stands for absorption, which means getting the drug into the blood and into the tissues. D is distribution, getting the drug to the right place in the body so it can do what it's meant to do. But M is metabolism, which deals with the reality that drugs are changed once they're put in the body. They are metabolized, and we have to be aware of how that metabolism happens and how it affects the active form of the drug. E is either excretion or elimination, and that goes to an understanding of how drugs are removed from the body. And T is toxicity, which refers to understanding the undesirable effects, what we know as the side effects of the particular drug. So those are, this is the guiding principle, and we'll be talking a great deal more, especially in the pharmacokinetics section, about ADMET principles. Okay, we're really looking with drug solubility at this issue of absorption, of getting the drug into the blood and into the tissues. Okay, as you should know by now, structure determines properties, and so far we've really been doing a lot of focus on drug structure. We've been looking at organic chemistry with a very specific eye to functional groups. Being able to recognize the functional groups in a drug molecule is critically important. The structure determines the properties and the activities. So what we're really doing here is what's known as SAR, stands for structure activity relationships, and SAR basically says that by knowing the structure we can often predict the properties and the activities of the molecule, or in this case it'll be the drug, and you'll get a chance to do that multiple times in this week's activities and labs. Okay. QSAR, which we have several, we'll spend several weeks doing, stands for quantitative or mathematical structure activity relationships. Yes, the SAR stuff that we're going to be doing and have done actually a little bit of already do involve math, but the quantitative structure activity relationships are highly mathematical, and we'll spend several weeks looking at those mathematics. Okay, determining drug solubilities. As I've alluded to already, drugs must dissolve to get into the blood and into the tissue. And the problem is that there are two environments in the body. There's an aqueous environment, which basically means a water-based environment. You basically are a large glass of water. Okay, most of your body consists of water, and that's the primary environment into which we're putting drugs. Okay, water, as you should know, is a polar molecule and capable of dissolving other polar molecules. Like dissolves like, so polar dissolves polar. But your body also consists of an awful lot of lipid structures. Lipid is another word for fats, and lipids are nonpolar molecules which are capable of dissolving or being dissolved by nonpolar molecules. So once again, like dissolves like, polar molecules dissolve polar molecules, nonpolar molecules dissolve nonpolar molecules. So you're really dealing with a chemical laboratory, which is your body, that really has these two environments, this aqueous environment and this lipid environment, and that's what makes this, what we're doing with drug solubility, so difficult. Okay, here's a picture of, sort of a cartoon picture of what we're dealing with there. The drug is the little green dots, and the idea is we've got to get this drug into the organs and the tissues, and it may get there by starting in the blood and then get disseminated out. So we may, A stands for absorption there, and we may transmit the drug from the blood to the organs and the tissues. Notice that some of the drug goes into the organs and the tissues, some of it may go into the brain. The CTZ there, by the way, stands for chemical transport zone. That's a brain specific function. We'll not talk about that now. We may talk about it later. But basically, the idea is that you've got these drug molecules that have to get absorbed and then distributed into the various either organs, tissues, or brain, depending again on what the drug is meant to do. Okay, so let's go back and talk now about why solubility is hard. Okay, and one of the questions we have to be able to ask is the chemical water soluble. And this is relatively easy to measure in the lab. Solubility is simply the amount of stuff that I can dissolve per some volume of water. And again, your body is think of it as a big beaker of water. When we put a drug in there, the drug is usually in a solid form, and that drug dissolves in the aqueous environment in your body, and we can measure that solubility pretty easily. And we use the abbreviation S subscript W, or S sub W, to represent the number that is solubility in water. And usually we report that in something like milligrams per liter or milligrams per milliliter. Okay, we typically, just to make the numbers even out and make things a little bit more even, we take the logarithms of the SW value and we report these as log SWs, and that puts our numbers on a little bit more reasonable scale. We don't have very big numbers or very small numbers. Log is a nice way to sort of even out the numbers a little bit. Okay, and the range is typically, and there you'll hear all kinds of values for this, but typically anything less than 0 for your log SW value is typically a reasonably good solubility in water. And again, there will be people who will argue about this range. Some people say it's a little bit bigger, it's a little bit smaller, but this is sort of a rough rule of thumb for, at least for this class. Okay, why is solubility hard? The next question you have to ask yourself, because we are in a dual environment, a dual system environment in your body, you got water and you got lipids, is the chemical lipid soluble? And again, that's relatively easy to measure in the lab. We have something called a partition coefficient, represented by a capital P, and that's a measure of the ability of a compound to dissolve in this mixture of lipids and water. So we measure the partition coefficient by how well a drug sort of intermixes within a lipid water mixture. Okay, so that's what we do. And you've encountered this term multiple times already in this course. We take the logarithm of this partition coefficient, this p-value, and it's known as a log p. Okay, and we've talked about this a little bit, and you'll remember this better as we go along. The higher the log P, that typically refers to the higher the ability of the compound to dissolve in a lipid environment, okay, which again is many of the cell membranes and other tissues in your body. So a higher log P means that the thing will dissolve more readily in a lipid environment, okay. If it has a negative log P, okay, we would call that hydrophilic, meaning it really loves water and doesn't like lipids. So that would be hydrophilic or lipophobic, whichever way you want to consider that. Okay, positive log P is again lipophilic. The higher the log P value, the more it loves lipids and wants to dissolve in them, and we would call that lipophilic. Okay, as we've mentioned already, the body is a complicated system. It's fundamentally an aqueous environment filled with lots and lots of lipids. Okay, you got membranes, tissues, cells, all of that stuff. And the body just doesn't behave as nicely as experiments in a lab. So we can measure solubilities and lipid dissolving ability in the lab, but your body, again, is a very complicated system that doesn't do as nicely as we can do in the laboratory. So the goal is to find the right combination of water-soluble and lipid-soluble compounds to get the drug into the body and to the right place where it'll do what we want it to do, cure the disease, make you feel better, whatever the case may be. Okay, here's a picture of hydrophilicity. So here notice I have a phenyl ring with a carboxylic acid on the top and another carboxylic acid on the right. Both those carboxylic acids are both hydrophilic. They love water. So they would, in an aqueous environment, as you see here with all the H2Os, this compound would dissolve quite well because it has very hydrophilic groups attached to it, in this case carboxylic acid groups. I hope you understand why we spent time getting you to identify functional groups. Okay, here's a picture of a cell membrane, very complicated picture. What you may see over on the far right-hand side is you have this bilayer, okay, this phospholipid bilayer. So you have a hydrophobic tails and you have hydrophilic heads. So parts of the molecule will, or membrane will repel water, some of them attract water. And so part of the problem is you got to get the drug through this membrane. You see over in the far left, there are things known as channels. In this case, a protein channel. We can actually slide things through a channel, but that's discussion for another time and place. Okay, but that's what a cell membrane looks like. Okay, improving water solubility. There's some ways we can do that, and this comes directly out of the textbook, so this is hopefully reinforcing the reading for this week. We can certainly add polar groups. The more polar the molecule, the more water soluble, because again, like dissolves like. Polar groups that ionize are better than polar groups that don't ionize. And some of you may know what ionization means, some of you may not, but here's an example, carboxylic acid. So C double bonded oxygen, OH, okay, will ionize or lose a hydrogen ion to form the ionized form, C double bond O, O with a negative charge on the oxygen. So in this case, this is showing that the carboxylic acid is ionizing, okay, to form the COO minus and a hydrogen ion, okay, and that's, so polar groups that do that, like carboxylic acid, are much better for making the substance water soluble than those polar groups that don't ionize quite as well. And we'll talk more about that a little bit next week. Okay, improving lipid solubility, what would you think? Sure, we add nonpolar groups, okay? The more nonpolar the molecule, the more likely it will dissolve into lipids. Once again, like dissolves like. And there are a variety of good choices. Some of these are in the textbook, some of these were elsewhere. Adding methyl groups, which are CH3 groups, makes the molecule more nonpolar. Adding fluorines, adding chlorines to your molecule will make these things nonpolar. And so those are some choices, and there's lots of other choices that you can make there in terms of making the compound more lipid soluble. Okay, improving water solubility, there are 3 general methods, and again, this is basically right out of the chapter reading for this week. We can change the drug into a salt, and again, some of you who have had a little bit more advanced chemistry or a little better chemistry will understand what this means. So a salt is an ionic compound that contains a positively charged metal ion or cation and a negatively charged anion. So an example would be changing a propanoic acid to a sodium propanoate. So here on the left I have propanoic acid, and make sure you can understand why. I've got 3 carbons, that's propane, and I've got a C double bond O OH group on the end, and that's a carboxylic acid, so we would call that propanoic acid. And what can we do there? We can ionize or remove the hydrogen from the acid group there, and now we have a charged acetate ion, or propanate ion, and we can add a metal sodium to it with a plus charge, and that now is what we call a salt. And we'll spend some time next week talking about acid-base chemistry, and that'll either be a review or an introduction to this kind of chemistry if you haven't had it yet. Okay, we can add water solubilizing groups, okay, like again, carboxylic acids. We can add phosphate groups with their -2 charge. We can add sulfonic acid groups. There's one too many P's there in sulfonic, I think. That's an S double bond O, O2H group, okay, so we can add those in there. We can add a bunch of polyhydroxy groups, so we can add a bunch of OH groups to improve water solubility. We can add ethers. In some cases, we can actually even add a sugar to a molecule, and that will help improve its water solubility. So there are specific functional groups or types of molecules, parts of molecules, that we would call water solubilizing, and we'll look at this a little bit at the end of this podcast. Okay, we can change the form of the drug. We can add co-solvents. We can turn them into colloids. Basically, surfactants, micelles, liposomes. Basically what we're talking when I say the form of the drug, we can change how this drug is dispensed to the patient. How do we physically get the drug into the body? Is it in pill form? Is it in a liquid? Is it a colloid? Do we mix it with some other solvent? So changing the form of the drug really refers to changing the dosage or the dosage form of the drug. And we're not going to talk about that anymore in this podcast. And I also did not have you read that section of the textbook. We'll get to that later when we talk about actually making drugs. Okay, emulsions is another one. Okay, so all of that talks to a little bit about improving drug solubility. And the lab for this week and the web assigns and the readings are all related to this issue of how do we make drugs more water soluble or more lipid soluble. I want to talk about one other. So all of this determines how these drugs get into the blood and get into the tissues where they need to be to cure the patient of whatever disease he or she has. But I do want to talk about one special uh, type of membrane here. And if you look on the left, what you see there is a sort of a cartoon picture of a typical capillary or typical blood vessel. So basically you have a cell membrane. It's got some openings in it, okay? But fundamentally the drug has to absorb its way through through that membrane to get into the cell to do whatever it is it needs to do, okay? The brain, however, has that same type of capillary structure as you see there in the picture, but it's also surrounded by another layer of a very protective, very tightly packed membrane called the blood-brain barrier. And the blood-brain barrier, or the BBB, does a very good job of screening out drugs from getting into the brain. And why was this a good thing? Well, there's an awful lot of drugs that you don't want in your brain because that's a pretty important region of your body. You don't want the neurons and the all the different components of the brain being affected heavily by taking different types of drugs. Now, there are certainly drugs like aspirin that are designed to cross the blood-brain barrier and get into the brain. Certainly hallucinogenics, many of the illicit drugs, morphine, heroin, ecstasy, crystal meth, those drugs give you highs because they're very good at crossing the blood-brain barrier and getting into the brain and messing things up there, and that's what makes you gives you the high that you get from taking those kinds of medications. Alcohol is the same thing. So you're going to hear a lot about the blood-brain barrier as we go through this course. Okay, and here's a nice picture. On the right-hand side this time is just a regular old capillary, just a regular blood vessel. And then on the right, excuse me, on the left, you see a better picture of the blood-brain barrier. So what you should be able to see there is the vessels are very loosely protected, but the brain is, we really do protect things in the brain. We really want to try to keep things out of the brain to the extent possible. Okay, there are a whole number of ways to predict drug solubility and in the lab, and you've already experienced a little bit of this. Okay, there's lots of things we can do computationally to try to predict solubility, but I want to introduce a pretty rough or pretty crude way of predicting drug solubility. And the answer you're going to get out of this method is not a number. You're going to get oh, this drug is probably not soluble, this drug may be slightly soluble, or this drug is soluble. And again, this is solubility in water. Okay, so if you look down in this chart here, down the left-hand side, we have alcohols, we have phenols, we have ethers, we have aldehydes, ketones, amines, carboxylic acids, esters, and amides. Okay, and what this chart is saying is that, and I'll start with alcohol, and we'll do a couple examples, is if the drug only has one of these functional groups in it, then it is capable of dissolving 5 to 6 carbons. So let's do an example, and that may help a little bit. So let's take a look at terbinafine over here on the right-hand side. It's an antifungal agent. Okay. The first thing you have to do to predict drug solubility using this method is count the number of carbons. Okay, and you simply have to count them 1, 2, 3, 4, 5. In this case, they've done that for us. It has 21 carbons in it. Okay, so for this to be soluble, I have to be able to dissolve 21 carbons. Well, how many are capable of being dissolving? So what you do now is you look for a functional group. And I look at this molecule, I see phenyl groups, I see some alkenes, I see an alkyne, a triple bond, I see a bunch of alkanes, but none of those are on the list under my list of water solubilization of functional groups. The only thing that appears on my list there is an amine. So if you see that nitrogen there, that's an amine. Okay? Being that that's the only functional group that I see in this compound that is on this list, okay, how many carbons is it capable of dissolving? Well, it can dissolve either 6 or 7 carbons. I'm going to take the higher number, and I'm going to say the amine is capable of dissolving 7 carbons. Well, that means 7 of the carbons out of the 21 are going to get dissolved, okay? And that's not enough. For this thing to be soluble, we really need to be able to dissolve almost all of the carbons. Now, we're not really dissolving the carbons. Basically, we're saying that the carbons are come apart in solution and they go somewhere else. So being how I have 7 out of 21, I would predict that this molecule is insoluble in its neutral form. Okay. Now, it turns out, as you see the note there, if I remove some parts of it, if I ionize it, I can actually make it soluble, but in the form that you see it here, if you were to have to predict this, I would predict this as being an insoluble drug. Okay, so let's go take a look at another. Okay, and it's an empirical method, again, a very rough estimate, and you count carbons and you compare those to water solubilizing effects. Okay, all right, let's look at one that's a little bit more complicated. Okay, once again, I count carbons, and I didn't count them, but the person who did this example did, and they're telling me that I have 18 carbons in this particular compound. That's an 18C atomer, okay, is how we would say that. And this is a beta blocker, a drug that helps people who are having chest pain, heart attacks. Okay, and I have multiple functional groups here. Okay, so if I look at this, I see 1 ether, I see 2 ethers, I see an alcohol, and I see an amine. Okay, so I see 1 ether, I see a second ether, I see an alcohol, and I see an amine. Okay, and now I look at my chart, and because I have more than one functional group here, for every ether, okay, I count 2 carbons that'll dissolve. So I have 2 ethers, that adds up to 4. Okay, I've got 1 alcohol, I'm going to consider that to be 4 carbons worth. And I have an amine, and that adds up to 2 carbons worth. And I always pick the higher number. If I have a choice of 1 or 2 numbers, I always pick the higher number. So that gives me a total of 10 10 things that will dissolve carbons, 10 out of 18. That's not enough to consider this thing to be soluble, and so I would consider this drug to be not soluble in water. Okay, and here are some examples for you to practice. So the first molecule on the top left, I count 9 carbons. I have 4, add up 4 carbons that can be dissolved, 4 out of 9. And sort of a rule of thumb that I use, and this is just my rule, you can make up whatever rule you want, but if it's less than 50% of the carbons that are dissolved, I would call it not soluble. If it's somewhere between 50 and 75%, I might call that slightly soluble. And if it's greater than 75%, I would call that soluble. So if you look, the 4 out of 9, that's less than 50%, so I would call that not soluble. The next one down, I count 13 carbons, and out of that I can dissolve 6 of them. 6 out of 13 is less than 50. I call that not soluble. The one on the top right, there's 19 carbons there. I can dissolve 13 of them, and I would call that slightly soluble in water. And the last one, the really big one down the bottom right-hand corner, I have 21 carbons total. I can dissolve 11 of them given the functional groups that I have there, and that's about 52% dissolving rate, and so I would call that slightly soluble. We're going to give you some of these on the web assignment to see if you can do. You may need to show up at an office hours tutorial to get a little help on these. Okay, the last thing I want to talk about, and there will be some web assign on this, is a little bit in organic chemistry about something called stereoisomers. Okay, it turns out, and you'll read about this in the chapter reading, which is why I wanted to present it. Okay, it turns out that a molecule can sort of have two configurations. Okay, and we call those R, and sometimes we indicate it with a plus sign, and an S configuration or a minus sign. I'm going to be doing a separate tutorial on how to designate stereoisomers because it's pretty important in drug chemistry. Okay, each of the two isomers there, and you're only going to have two, okay, so they're basically stereo images of each other, is called an enantiomer. Okay, so I have two. I have an R enantiomer and an S enantiomer. And if I have a solution that has 50% of the solution is R and 50% is S, I would call them racemates. Some of you may have seen that with when you were building molecules in the organic chemistry drug design tool. You might have seen these words pop up. That's what the E stands for when you see that in the organicchemistry.org. So a racemate is a 50-50 mixture of enantiomers. And again, what I'm going to do for this stuff here is put together a separate podcast that will be posted very soon that will do a tutorial on how to recognize and how to identify stereoisomers. So for this week, the reading is chapter 3, pages 37 through 59, which talks about drug solubility. You're going to have a mini lab to try to identify 10 drugs for their stereochemistry and also identify the solubility for 5 other compounds. That's a little mini lab that'll actually be on the web assign. The lab this week will be obviously drug solubilities, and it'll be very much like last week's lab where I give you a case study scenario, you have to solve a problem, and you have to reply, communicate your solution via memo form. Okay. Any questions? We'll see you in the tutorial sessions. Thank you very much.